Welcome, everybody. My name is Tommy Source. I'm Assistant Director of the Purdue Institute of Inflammation, Immunology, and Infectious Disease. And I want to welcome you all to another Purdue Lecture Hall series. Today, we have the pleasure of honoring our guest today, Conrrad Nicholls from the laboratory of Dr. Richard Kuhn in the Department of Biological Sciences. Conrrad is specializing in understanding how a particular type of viruses are assembled. And welcome to the show. We're so excited to hear you talk and tell us a little bit about your journey into science. And as you know, Conrrad, this is an opportunity for anybody who is interested in research, interested in science and what it takes. And so we welcome high school students and undergrads to come and hear your story and kind of take a little bit out of it and say, wow, what can I do with my hands as well? So thank you for coming here. Thank you for teaching us and inspiring us all to follow in your footsteps.
Oh man, well, I hope I can do that all justice. I appreciate the intro, Tommy. That's very nice of you, and I'm very excited to share my work and a little bit about my background. So I'm happy to be here. Thank you.
Well, thank you. And I guess, you know, you can take it away because it's your lecture, it's your presentation. I'm going to turn my camera off and mute myself until you're ready for maybe a question or two, if we can do that.
That sounds great. And if anyone along the way wants to pop in and ask a question, please stop me. I'm happy to answer stuff along the way. So that's I feel like that always helps the flow of things so people don't forget or have to go back and slide. Please hop in at any moment, but thank you. All right. Well, thank you so much again, Tommy, for the introduction, and thank you all for being here today. I'm excited to share with you all my work involving flaviviruses. But the title of my talk today is Building a Virus, Identifying Connections Between the Core and the Envelope Proteins of Flaviviruses. But before we get into that, I want to go ahead and, oh no, I'm frozen already. We're off to a great start. Let's see here. Let me try and move. There we go. I just got pressed a little bit harder. So before we get into all of that, I want to walk you guys through my background and some biography for myself and how I slowly made my way out east. So I started by being born in the beautiful state of Hawaii on the island of Oahu in the city of Honolulu. And unfortunately, it is very expensive for a new family to kind of start growing and developing. So my family did have to leave the beautiful shores and white sandy beaches of Hawaii. But I was there for about 2 years, and it was a beautiful place. And I don't remember much of it, but plenty of pictures. And we do go back often to visit. But we left Hawaii, and we actually went to Las Vegas, Nevada, where we had many family members who actually lived there. And my parents were able to get more established, and I was able to gain a little brother along the way. And so he was born in Las Vegas, and our family started growing piece by piece. And it was nice to kind of just develop and grow and learn new things along the way. But I reached my stride in when we moved to Kansas City, Missouri, where we had more family there. Then in Las Vegas, and a little bit more established. Cost of living was a bit better. But I actually lived in Missouri from about the age of 7 all the way through graduating high school. And so that's where I consider home. While Hawaii is the most, prestigious place, it's very fun, it's beautiful, everyone wants to go visit. I like to claim that as home, but home is Missouri. But what brought me to Indiana was actually football, and I'll get into that here pretty soon. But I was able to attend Indiana State University. I was a Sycamore, and it's just down in Terre Haute, Indiana. And I've unfortunately gone from white sandy beaches to cornfields all the way in Indiana, but I've enjoyed the trip along the way, and I'm sure there's many more fun memories to make going forward. But that's kind of in a nutshell, a little bit about my background, how I got all the way out here to Indiana. If you guys have any questions about any of those cities, happy to answer at the end here. But like I said, my undergraduate institution was Indiana State University. This is a smaller campus, much smaller than Purdue, of course. This only has about 20,000 students, including grad students. And so it's about half the size to about a third the size of Purdue. And it only takes about 15 minutes to walk across the whole campus, but it's very beautiful. has a lot of fountains and beautiful architecture throughout the campus. And it was a nice, kind of cozy place to be. But as I said, I did go to the university to play football. So I actually played defensive end, and I was on the dark side of the football, as we like to say. But this gave me a golden opportunity to pursue higher education in a manner that I would not have been able to participate in had I not played some sports. So this was a great opportunity for my family and myself to kind of further that education and to gain a foothold into maybe a higher socioeconomic status. And so I'm very grateful for this opportunity, and I think this is something fairly unique to my experience going into grad school, of being able to utilize some physical skill sets for myself to propel me into higher education. And I know a lot of students around the globe that don't really have the means to go into higher education, and sports is often a way for them to do that. And so I think I'm very blessed and very thankful for the opportunity to have played ball. I got to play for 4 years, and it was the time of my life. I missed it all the time, and I wish I could go back. And I wasn't able to watch football for quite some time after I left my undergrad, but it was a great time. And I learned a lot of good things. I learned a lot about leadership. I learned a lot about communication, and especially teamwork. We had guys on the team that were, from inner city Atlanta all the way to, backfield corn woods, Indiana, and everything in between. We had guys from Canada, we had a few guys from the South Pacific, a guy from China. And so it was nice to kind of see all those diverse backgrounds, see how we can all bring a specific skill set to the table and have a unique and coherent goal to chase after, which of course was winning games on Saturday, but it was bigger than that. It was trying to come together and develop chemistry among the team and to really put our differences aside for a common goal. And I think that's something that is I will carry with me throughout the rest of my life, and something that I encourage everyone to try and develop to the best of their abilities, no matter where they come from or whether they play sports or not. And some of my great colleagues here, I've also learned great teamwork, and they didn't play any sports, but it's a universal, I think, characteristic that can help in many different ways. So enough of my soapbox there. Football was fun. I had a great time. But also while I was in undergrad, I was able to participate in undergraduate research. And so I've always had a fascination and love of science. And that's what I wanted to be since I was a little kid. I wanted to be a scientist. Actually, I wanted to be an astronaut, but then I got way too big for a lot of the simulators. I'm technically 6'5". So a lot of the NASA dreams kind of went out the door with that. So they have like an average height of, I think, like 5'8". But instead, I wanted to be a scientist. And so I thought, you know what, this is something that I've always wanted to do, something that I enjoy doing. I was majoring in biology at the time. Whereas a lot of my other teammates were in business, excuse me, in marketing, or even some in hospitality. Not knocking those fields, but that's the main kind of sports agenda, is you kind of come in, you get a business marketing degree, and you leave. I want to do something different. I wanted to do science, and that's something I could see myself doing even after football. And so I was able to participate in research both my junior and senior years. And I got to work with, it was the first time I got to work with viruses, but also bacteria. Other than this, it was mostly genetics work. But I got to work with the Streptococcus pyogenes bacteria. Now, this is a gram-positive bacteria, and it's usually found in normal skin microflora. But if it gets out of control, it can cause some pretty bad disease. And the way that it does that is it harnesses high hyaluronic acid. It produces it, and it's something your body naturally produces. And it can use this HA, or high hyaluronic acid, to hide from the body's immune system. But the PI I had at the time in my undergrad research, he was really interested in how prophages, or bacteriophages, DNA or RNA, can in fact help the bacteria to evade the human immune system. How can these prophage genes actually help in the hiding process of the Streptococcus pyogenes bacteria? And so what we look for with these things down here are called prophages. If you can see my cursor right here. Now, prophages, like I said before, are some phage DNA that is integrated into the bacterial chromosome to become a prophage. Now, a lot of viruses use techniques such as this to kind of hide from the immune system, such as like HIV in humans, to then cause infection later on. But some bacteria, especially bacteria such as Strep pyogenes, that are capable of causing major pathogenesis in humans, can actually use some of these prophage genes to their advantage to heighten their ability to produce more high hyaluronic acid and even some other unknown genes and methods of hiding and evasion from the immune system. And so we were actually, my colleague and I were able to take a new strain of Strep pyogenes. We were able to extract its DNA and sequence the entire DNA and annotate it according to sequence alignments and using blast sequences, a bunch of fun little things you don't have to worry about right now. But we're able to find that, okay, look, there's a few genes here that we can actually conclude help these bacteria from a prophage cause more harm and more pathogenesis. So it's a great kind of entry point into how viruses and other pathogens such as bacteria can work together to kind of make each other stronger. And we're still trying to catch up. We're still trying to figure out how they do that. And so it's a nice kind of, like I said, entry point into studying this kind of idea. And I was fortunate enough to be able to publish this genome in a draft genome sequence publication. And I was able to present my work at the National Conference for Undergraduate Research. And this was a great kind of eye-opening experience for me specifically because at the time I was just doing research. I thought I'd kind of go straight into industry, maybe potentially go in to be a laboratory tech. I didn't really have grad school as the ultimate goal. I honestly didn't know enough about that. But going into NCUR and going into this publication, I learned so much about what the field of science looked like, how I was supposed to go about a career path. And it really helped to open my eyes to see that, hey, this is something I really do enjoy. And it's something that I would encourage everyone to pursue if they can, obviously. And so this, of course, was a great stepping stone to then take me to Purdue University. And I was able to get into Purdue University, and I'm here still, which is awesome. And to kind of transition from my undergrad institution to Purdue University, the first step I took was utilizing the Alliance for Graduate Education and Professoriate, or AGEP program. Now it's nice because once you graduate from undergrad, there are several different programs to where individuals can join higher education or a graduate program ahead of schedule. So most people start in the fall, like in August, September. But for the AGEP program, they can bring these students, they're called bridge students, directly from undergrad right after graduation in May, and you can have an entire semester of graduate school research during the summer, prior to even starting graduate school. So it's a nice, again, another foot in the door, another way for me to see if this is something I did truly want to do. And I was able to participate in that. So in 2016, I was able to participate in the AGEP program, bridge program. I got to come here. I actually joined the lab that I'm currently in now. So it all came full circle. But this has been a great experience. This was a great experience, to understand how grad school research actually worked. So in addition to that, I was able to participate in a bunch of other programs here. So kind of jumping ahead a little bit in the timeline of everything. But something I wanted to share with you all here was that while in grad school, there are many different opportunities for which you can participate in for networking or for skill sharpening or for just purely interest. And there's so many different directions you can go. It's hard to say no to some things, but it's worthwhile to explore all the options in front of you. So that's exactly what I wanted to do. I wanted to take advantage of this wonderful opportunity I had to be in grad school. And so the first thing I did was I participated in Pulse Science in Schools. This is a program that allows graduate students to go to a elementary school here in the greater Lafayette area. And we teach second through fourth graders kind of the basic concepts of science, how to build a hypothesis, what the scientific method looks like, how you can think about certain things, and if you have a question, how to test those questions. And we would, of course, build like lava lamps, and we would extract DNA from strawberries with them, and it was a blast. But it kind of gave me an extra glimpse into my own childhood, of saying, man, this was something that I loved doing as a kid, and I'm able to kind of give back, and everything's able to come full circle with new potential scientists. And so in addition to that, in 2017, I thought, okay, what else can I participate in that will help me succeed and give me kind of an edge on the competition? And the great thing about Purdue is that it has a dual degree program that allows you to get both a Master of Public Health as well as a PhD. And so some, of course, programs don't really gel well, but I thought, you know what, with being a virologist, someone that studies, pandemics, someone that studies pathology and virulence, what about the public health side? How are policies developed? How can you integrate what I do at the bench side to the public sector? and how can I get my information to the lay person in a way that they would understand that. So this whole master's program really helped me develop those skills and learn how to become a better communicator of my science with the lay public. And so this was great. I was actually able to complete this master's degree this past December. I'm still chipping away at the PhD, but we're almost there. So in addition to this, I was able to participate as the president of the Polynesian Cultural Club of Purdue University. It's a mouthful there. But in the PCC, it was great because coming from Hawaii, being raised in Polynesian culture and understanding the different traditional dances and the food, it was really awesome to share all of that with the people here in the greater Lafayette area. We were able to host 2 different luau's, which are, large celebrations and feasts with lots of good Polynesian food. With a bunch of people here, we actually had 2 events, both had about 500 people in each. And it was a great networking experience, but also just a great time for me to share my culture and heritage with people who might not have been exposed to it. So that was great. And then finally, I was able to participate in a program called Gadget. It was the Graduate Assistant Development and Guidance Educational Team. Now, that's another mouthful. Apologize for all the acronyms. Scientists and clubs on campus do love their acronyms. But this was a great opportunity because I was able to take all those skills I developed in undergrad through football, through teamwork, and apply that kind of pedagogy to grad school. As TAs, some of us have great communication skills, some of us don't. But what we need to understand is that the goal, the unifying goal, is instead of winning games, it's to make sure that the students you're teaching get the most out of your courses. And so that's kind of what we went into. I was there with 3 other graduate students and then also a PI who was the mentor for this program. And we developed an entire curriculum to try and help TAs become more professional in their communication abilities, but also a little bit more understanding in the idea of cohesive teaching. So that was a great opportunity again, sharpening those skills, learning how to become a better communicator myself, learning how to then push those boundaries of what I was comfortable doing with my colleagues, and how we communicate with each other, how we take that information to a layperson, in this case, to students. And so these were all great opportunities that I've had along the way, and they've kind of helped me develop into the person I am today. And so with all that said, that's a little bit of my background. I'm sorry I'm so long-winded when it comes to that stuff, but it really makes me happy, and I'm excited to share that with you. So with all that, though, I want to kind of get into my research and just the research I'm doing in general. So the first question I want to pose, and this is, of course, a rhetorical question, is what is a virus? So a virus, in a nutshell, can come in 2 different forms in its most basic idea. One is a non-enveloped virus, and this is going to be important in a little while, and we'll get to it. But a non-enveloped virus is something that contains a protein shell or a capsid, something that holds everything inside, and it's usually just protein. There's no lipids in what's called a non-enveloped virus. And then inside that capsid is genetic material, such as DNA or RNA. And the RNA can come in a couple different forms. It's single-stranded or double-stranded. If it's single-stranded, it's positive or negative sense single-stranded. That's not important, but just know there's a bunch of different ways that genetic material can be packaged. But then we also have what's called an enveloped virus. Now, an enveloped virus contains the same thing, a protein shell. In this case, it's inside of the envelope, and it's called a nucleocapsid because it's kind of like the nucleus of a virus. This, of course, still contains the same genetic material, DNA or RNA, and the different forms of RNA. But then what makes this different from the non-enveloped viruses is that it contains a viral envelope or a lipid bilayer. And so this lipid bilayer is oftentimes garnished from the cell that the virus is grown in. And we'll get into that here in just a minute. So don't worry too much about the ideas here. But this viral envelope contains a lot of the same lipids as a host cell. It can be used in camouflage. But most importantly, it helps to embed in what are called envelope proteins, those little blue spiky things you see around the edge there. Now, envelope proteins serve many different purposes. They can act in virus attachment. They can help with virus virulence. The way that those envelope proteins are presented can help them stay away from immune cells. But these 2 types of viruses are just the main ideas of what a virus is, but there's so many different ways in which these simple machines can be produced, and they can cause disease. Getting into that here, first thing I want to do is talk about there's many different types of viruses. So what we saw in the last slide, those 2 things looked pretty similar, but one just had an envelope with spiky things on it. However, what we can see here is that up in the top left corner and in the middle of the diagram here, we can see 2 non-enveloped viruses that just contain protein. So we have the mastodinovirus, and we also have papillomaviruses, enteroviruses, rhinoviruses, such as the common cold, and even rotaviruses. Now, these are non-enveloped, and they're just protein-based, but they do look similar in some ways, but they're just protein. But then when you throw in the envelope side of things, we have coronaviruses, herpes, we have smallpox, rabies, Ebola down there at the bottom left, and even hepatitis D and hantaviruses. Now, these are all very different. We have different morphologies, they have different functions, they are built in different ways. And this is why it's so important to kind of understand that, okay, yeah, these two simple machines, non-enveloped, enveloped, can be presented or built in so many different ways, which gives them so many different functions that can cause so much disease. So with that in mind, I kind of want to talk about an analogy I like to use when it comes to why different structures matter. So I like to use cars, right? I love cars. I'm a truck driver myself. I love a good vehicle that is just different from the rest, but still useful. So what we have here is we have our McLaren on the left, we have a Toyota minivan in the middle, and then we have a Ford Raptor truck on the right. And they're all vehicles, right? They all have a similar function. Their main function is just to drive, but they're made with drastically different parts depending on the brand of the vehicle or the kind of driving that's required. So, of course, like a McLaren, that's for a racetrack, right? You want to go 200 miles an hour and you want to do it as efficiently as possible, you want a McLaren. If you want to take me to football practice, you probably need a minivan, me and all my little buddies. But myself, I drive a Ford Raptor. That's something for like off-road. And so it depends on the kind of driving required. And the viruses utilize a comparable approach. They use different structures or shapes, as you saw in the last slide, to exploit different aspects of an infection in their host. And so to kind of draw a better picture of that, I want to kind of paint a picture of the virus life cycle. What we have here on the screen is a very simple life cycle kind of diagram, but I want to walk through each of these different points and discuss how, in fact, they can play a role in specific viruses. So the first one is, of course, attachment. The virus has to attach to a cell, it has to be grabbed by the cell, and it has to be pulled in. So SARS-CoV-2, as we're all very unfortunate in understanding and knowing, utilizes an ACE2 receptor, as well as others that we probably don't even know yet. But the ACE2 receptor specifically can bind to the SARS-CoV-2 spike protein and pull that protein and the virus, into a cell. And so that's how SARS can be able to get in. Many different viruses use a myriad of different receptors to do that. Now, once in the cell, certain viruses start to do replication and translation, meaning that they start to replicate their genome. That genome then turns around and starts making proteins, which are the building blocks of the virus. And those building blocks then coalesce and form new particles. But during this process, certain viruses can actually go in and hide inside of the cell. They can take their genetic material, put it into the genomes of these cells, and wait to cause infection at a later time. Not quite sure how or why they do this, if at all. Why is a very anthropomorphized term, but what is the purpose for this hiding? And we're not quite sure yet, but it's very interesting. It's something that HIV does. It's not something that flavies do. It's not something that SARS does. So what is the kind of reasoning for this? But this is a unique specific section that a specific structure, in this case HIV, is capable of doing. Now, the next one we have is assembly. So once all the pieces are made, all the little building blocks of these viruses, all the proteins have been translated, they then have to come together to form new particles. And what we have here is an Ebola virus and a rabies virus. Now, both of these viruses assemble, they put all their pieces together on the plasma membrane of a cell. So if you think back to Biology 101, you have a nice little cell, circular cell, and has a plasma membrane on the surface. That's where all these little proteins are embedded. These viruses form on that surface. However, some viruses, such as flavies, they actually form in the endoplasmic reticulum, and other viruses form inside little vesicles. And so how the virus puts itself together and where it puts itself together is very interesting and often very useful when trying to understand how this all works. And lastly, we have how a virus is released. And so we have polio here, for example. This is usually a virus that undergoes a lytic exit or a lytic egress from the cell, meaning that it bursts the cell open and it releases all the particles at once. Other viruses, such as flavies, that I'm interested in, they actually will bud out of the cell using the Golgi, trans-Golgi network, egress. And so these little viruses will bud into the ER, they'll be trafficked through the Golgi, and then they'll slowly be released. And so they don't kill the cell when they're released. So all these different viruses use different mechanisms altogether to cause disease in their own unique ways. And so kind of coming back to this whole idea of cars, right? What we have on the left is this McLaren assembly line. It's a specific setup. It has unique machines, unique personnel that are capable of building that vehicle. Then you have a Toyota assembly line where you don't see anyone on the assembly line, but it's all automated, it's all machines. So the idea is, okay, what kind of tools, in this case, what kind of cellular components are being used to build the virus? Do they require hand assembly, like the McLaren, or are they automated? And in the virus's case, do they require unique enzymes, or are they hijacking regular cell machinery that is capable of just doing this on its own? And so it's not like the virus is maybe bringing in a unique enzyme with its own genetic material that is specialized for it. It's just using whatever's available. And so these are all important aspects because, okay, if we can figure out, all of these different pieces, even how the virus is shipped out, once it's assembled, does it get out through blowing up the cell, or does it get out through simple budding and where these cells are still alive so it can make more? The idea is, okay, in order to learn how a virus causes disease, it is essential to learn how it first builds itself. This is why we study the cellular life cycle and also study host and virus protein interactions. Now, knowing the life cycle can then allow us to design drugs that can interrupt specific or unique building processes, then taking this assembly plan completely offline. So without constant production, the virus will not be able to cause disease or spread. And so I know I kind of beat this dead horse a little bit, but the idea of an assembly line, of building a vehicle, is exactly the same when it comes to viruses. What pieces are made, how are they fitted together, and what is the purpose once it's off that assembly line? Can we find a specific area in that whole process we can unplug something, or can we disrupt a specific gear to stop that whole thing from happening? So this brings me finally to my research specifically, and these are with flaviviruses. So what we can see here are three structures of three different species of flaviviruses. We have Zika virus that maybe some of you have heard of, caused a global pandemic in 2017. We also have dengue virus. Now, dengue is something that is widespread throughout the globe. We'll get into that here soon, right here in the middle. And then we have West Nile virus. What we can see is that all three of these viruses, while they are unique in and of themselves, they have specific sequences, they have specific genetic material that is slightly different from the other. They have a very similar structure. So it's almost like a Ford Raptor versus Ford F-150. One is built strictly for off-road, one is a bit more fine-tuned to the highway. But they are very, very similar. You can probably swap out some parts and they'd be fine. But this is what's interesting is because flaviviruses come from, they cause a myriad of disease, right? So they're primarily passed on through mosquitoes. So in this case, the Culex and Aedes aegypti generas, or specific mosquito types. They're also passed on by certain ticks, and then also there's a bunch of unknown vectors. So a vector is something that can take a virus, or in this case, a pathogen, and pass it to another species. Now, flavies can manifest in a bunch of different symptoms. They can cause rash, they can cause fever, meningitis, encephalitis, which is the swelling of the brain, and also even hemorrhagic fever and microcephaly. So, I'm sorry, make sure oh, cool. So don't see the rest. Thank you so much. You guys be safe. I hope that you're able to survive that snowstorm. Everyone's clean and happy and nice and warm. So you guys take it easy. Bye. Anyways, so back to the talk here. So a lot of different flavies can manifest in a bunch of different ways. Now, the reason this is so important, the reason why I'm studying flavies, is because, sorry, I'm frozen again. Let me see if I can. There we go. The flavies are a global issue. So what we can see here from this map is that Zika virus is able to be detected in 89 different countries. Now this is pretty significant because 89 countries that have very low levels of reporting still report it means that it's probably much more prevalent than we can see. Now this specific virus, Zika, can cause microcephaly in newborns and, in fetuses that result in a low survivability for newborns, which is quite detrimental. Also, dengue virus infects 390 million total infections per year, with 100 million of those resulting in clinical cases and about 500,000 presenting as severe dengue fever or severe dengue disease. Now, Japanese encephalitis virus, which is another flavie, it results in about 50,000 severe neurological diseases throughout Asia every single year. And West Nile virus is currently emerging, meaning that it's currently growing in number out in Eastern Europe. And so this is quite difficult to combat because all these flavies are causing major issues. And altogether, there's about 3.1 billion people at risk of flavivirus infection every single year. So there is a deep and significant need to develop antiviral therapeutics capable of targeting ubiquitous mechanisms across most, if not all, flaviviruses. So to do that, again, we go back to the virus life cycle. We go back to those assembly lines of how these viruses are being built, which is try and figure out what is going on. What we currently know is that we know quite a bit about virus attachment and entry. We know quite a bit about virus encoding, and we even know quite a bit about virus maturation, how these viruses go from spiky particles to smooth particles. And that's not really important right now, but we'll get into it here soon. So we know a lot about that, but we don't know much about the flavivirus assembly process. So the primary question I'm seeking to answer is, how does the nucleocapsid core bud, or kind of push into the protein-rich membrane of the endoplasmic reticulum? I don't know why I can't say that word today, but the endoplasmic reticulum, to form immature particles. How does this process happen, right? So what we can see here is in this little diagram, I don't know if you can see my cursor, but what we have is a bunch of little capsid proteins and envelope proteins all kind of coalescing inside of the cytosol between the endoplasmic reticulum membrane and slowly pushing in to form these immature particles. But how does this process take place? What interactions are occurring? We're not quite sure. But before we get into all that, I want to kind of go through some of the flavivirus genome and polyprotein. So the genome, as I hinted at before, is a positive sense single-stranded RNA genome. Now, this genome encodes two different groups of proteins, the first being a non-structural group, which is all the NS proteins that you see here on the right side of this diagram, non-structural protein 1 through non-structural protein 5. And the function of these proteins is in replication, which meaning replicating the genome, as well as different protein machines, the assembly of the particles, and as well as immunoevasion. But then we have these structural proteins, and these are highlighted here in this red box, the capsid pre-membrane / membrane protein and the envelope protein. Now we're going to be especially talking about these and the transmembrane helices, so these little anchors inside of the lipid bilayer here, and how those potentially may play a role in virus assembly. So with all that in mind, we kind of go back to maybe there's a few different mechanisms that are currently hypothesized to take place that can maybe help in the assembly process. So one that has been proposed is the idea that this protein down here, this little dumbbell, this blue dumbbell-shaped protein, NS2A, is potentially involved in chaperoning or bringing in all these other proteins to these sites of assembly. So all this is taking place on a molecular level, and there's still maybe a little tow truck, this little NS2A protein, this little tugboat, pulling in a bunch of different proteins to the site of assembly where all of the interactions can take place and particles can be formed. That's all you need to take away from this picture here. Additionally, there could be some ways in which the same protein is capable of functioning in different ways to help with the assembly process. What we have here up in panel A is we have a bunch of different envelope proteins that are kind of congregating around the lipid bilayer of the eventual virus. All those are kind of just coalescing and forming because they're being produced by the ribosomes and all the other different molecules on the ER. But then also what you have inside the cytosol are these two groups of the same protein called capsid. Now capsid is a structural protein that was believed to only interact with the RNA to then form a ball of RNA in capsid, which somehow gets inside of an envelope. Not sure how that works, but some of the hypothesized ways in which this can happen is that maybe capsid, during its building process, retains something that we thought would be cut off, because there's this little helix that kind of sticks out from capsid that has what's called a protease recognition site in it, which is a cut site inside of the protein sequence. And we thought that this little helix would just be cut off because of that sequence. However, there's some data showing that maybe it's retained, maybe it's being held on. And if it is being held on, maybe it can have two functions. Maybe there's a group of these capsids that doesn't contain the extra helix and one that does. The one that does is capable of forming what we call a trimer of dimers, meaning that there's 3 dimers. And so a dimer is just 2 capsid proteins coming together to form a dimer. And then all those little dimers then form a trimer. So it forms this little kind of triangle here. Now this little triangle, we think, is potentially able to interact with either the helices or directly with the membrane, while the non-helix containing capsid is able to interact with the RNA. Basically, when they all function together and they all kind of coalesce, what happens is, okay, the non-containing capsid bundles the RNA. The capsid that does contain alpha 5, or in this case, the extra helix, is capable of bending the membrane and starting the budding process. That's pretty cool. We think that might actually be the case, but we're still trying to figure that out. But next, we have what we call asymmetric budding. This is when the nucleocapsid core, what we see here is this little purple ball, is somehow pushing into the membrane, but it's not doing it in a symmetrical way. And I'll get into why that's important here in just a second. But it can push into the membrane, and it can start to form this little bubble around itself, and it kind of pushes up against one side, and that's why, hence the term asymmetric, is not directly in the center. It is not directly outlined completely by the membrane. Instead, it is pushed slightly to one side, and it's asymmetric as a result. But what we see is, in the immature particles, which I'll get into in just a second, we see kind of this asymmetry, where it's pushed up to one side. In a mature virus, we see that it's kind of pushed back into the middle. So maybe these processes are all happening in conjunction with one another, where we have asymmetric budding occurring and some interactions taking place. But then during the maturation phase, when the particle goes from spiky to smooth because of different protein interactions and different protein changes and movements during the maturation process, that interaction is lost. So maybe there's something in here that can detach this core from the lipid bilayer, and we're still trying to figure that out. But going into the actual data here, so just there's something here that's a very beautiful picture, but this is what's called a symmetric reconstruction. It's where we take a bunch of different pictures of viruses that we've been able to purify, and what we do is we do an average of all of those little pictures, and then we put those averages together, and we get a structure out of it. Hence, what I said before, it's like taking all the pieces of a car, laying them all out, then finding a bunch of different ways in which they build each other, and then taking all the different possible pictures of those, overlaying all of them, and finding an average, and then getting what that car should look like. That's the way we usually do it, but what's been currently being done in our lab is what's called asymmetric reconstruction. This is not using any type of symmetry imposition, which means we're not giving the computer program anything to look for. We're saying, look, take what you have and figure it out on your own. And this is what we're able to find, is that there is this pushing up of one side of the core to the actual membrane of the virus. But the question still remains, oh, excuse me, I'm sorry, I had a few things here. The question still remains, are there any interactions taking place between the nucleocapsid core and the envelope? That is still the question. So can these interactions be perturbed? Can I mess them up? Can I throw a wrench in the machine to stop it from happening that can prove that this interaction is taking place? So some recent structural studies provide some more information on this potential interaction between the capsid protein and potentially the pre-membrane protein and the envelope protein. So what we have here on the left is that smooth particle that I've talked about before, the mature Zika virus. This is the structure of it that we've gotten in our lab. And over here on the right is the immature Zika virus structure. Now, it's getting a little bit more in-depth into the science of everything, but for now, just worry about the differences between the two. It's like those little pictures we used to do, when we were kids, right? What is different? What's different here? What we can see is that for the most part, they're fairly similar. What we see is the envelope protein transmembrane helices existing right here in kind of this little red circle are both present in both forms. The lipid bilayer, shown here in this green kind of line that goes around, is here in both. And the nucleocapsid core, shown here in purple, is also in both. However, there's this residual density that, what I mean by residual density is that there's this leftover something that is not seen in one of them that is present in the other. And this little piece right here seems to fit the capsid protein. What I mean by fit means that there's just a bubble of something there in the picture. We're not sure what it is. But when we take the structure of the capsid protein and put it into that little bubble, it fits almost perfectly. And so is this a part of the entire virus that needs capsid to fit in it in order to form? So that's the idea, right? So going a little bit further, what we have here is the mesh structure. So this is kind of the zoomed in version and getting rid of all the noise of the image you just saw. And capsid protein is sitting in that little bubble with that little residual density. But what is right on top of it? We're not quite sure what is right on top of it. So the idea was, okay, cool. Is this the central spike? Is this something that is sitting on top of it? No, it doesn't really fit very well. Well, how about the adjacent spikes? Maybe, but it's too noisy. So let's get rid of the noise Let's focus only on their transmembrane helices, and let's get rid of everything else and just put those into the structure. And what we can see is that the helices themselves, those little anchors that keep those big proteins anchored into the membrane, are directly above capsid. They are right there, ready to be used in some way that we're not sure is necessary. So the idea is, okay, I have this idea that maybe those specific helices are interacting with capsid. So how am I going to test this? This is where good scientific methodology comes into place. This is a hypothesis building. Now, I'm hypothesizing that the assembly, the building of those specific infectious flavivirus particles is dependent on specific interactions between the capsid protein and some specific amino acids in the envelope protein transmembrane helices. So that little part that's sticking down on the membrane, some type of interaction may be taking place right there. So what I'm doing is I'm mutating specific amino acids in the envelope proteins to see if I stop virus production. I am then looking to see if these mutations kill the virus or make it very difficult for the virus to proliferate. This would suggest that these amino acids are essential for virus assembly. And then also, this could be a new druggable target capable of stopping the spread of flaviviruses. The idea is, okay, I'm going into the engine of the virus, in this case, and I'm trying to take a few bolts out, or I'm changing one. I'm putting a different size bolt in where a different one was supposed to be and seeing what happens. I'm changing a piece of it to see how it affects the whole. And so to do this, and I think I'm a little bit low on time, so I'll tell you what, I will actually, Tommy, I'm going to go ahead and fast forward just a bit here. Go ahead and way too much for this.
No problem. No problem at all.
So what I'm doing is, actually I have these 3 slides, that should be fine. So to do all this, I'm using what are called RVPs. These are reporter virus particles. These are particles that give me an idea of how infection is taking place. What I do is I use a specific plasmid, in this case a circularized DNA piece that contains a reporter gene, in this case GFP, and then a bunch of those non-structural proteins that help in the development of the virus or in the replication and assembly of the particle. But they come from West Nile virus instead of from Zika, which is what I'm interested in. But then I have a secondary plasmid, one that comes specifically from Zika that only contains the structural proteins. I put both of these plasmids into cells, and then I look to see how it changes. So what I do is I build, in a sense, pseudo-infectious particles, particles that are only capable of infecting a single round. These contain Zika structural proteins, but a West Nile virus replicon, in this case, a West Nile virus replication machinery. Now what this does is it allows me to use fluorescent cell counting. I can check to see which cells are green, and if they're green, that means they've been infected. And then it helps me kind of reduce my screening of mutants down from 9 days to only 5. And so to kind of build a picture for you guys, what we have over here is this inverted tripod, meaning that this is the envelope proteins that are kind of sticking out of the top here. We have our pre-membrane protein transmembrane domain, the little anchors that hold it into the membrane. And we have our E protein transmembrane domain, the E protein anchors. And we have our capsid protein that's hypothetically interacting. What I did was I identified specific amino acids that could potentially be interacting because these are the closest things to the capsid protein, what we're hypothesizing is the capsid protein. And so I'm going to change them. I'm going to take each one of these specific amino acids. So what we have here is a leucine, for example, at position 146. I'm going to change that to something else and see what happens. And so what I did was I designed 86 mutants containing single point mutations, meaning that a single swap was made, or a major insert ranging from 2 to 5 amino acids. So I could either put in right in between G147 and S148. I added 2 amino acids, or I added 5, something in between there. And then what I saw was I did this both in dengue and in Zika. And altogether, my current data suggests that changes to these amino acids stop virus production in some of my mutants. And so far, I've been able to isolate 25 different mutants that inhibit virus growth. So I know we're short on time, so let me kind of get through all of this here and go straight to my conclusion side. What I have is that, okay, having understood now, going back to this car analogy, how my McLaren GT3 is assembled, understanding its virus life cycle, I was able to pinpoint a specific area along the assembly line to be tested for weaknesses, virus assembly. I then, after changing out some of the components in the engine, mutating specific residues, I found a way to keep it from driving. I found a way to keep the virus from making itself. I then used this approach across the board, and I'm nearly able to conclude, that flavivirus proliferation can significantly be reduced with a few single point mutations in both the E protein and the PRM protein, giving us new targets for drug research and potentially, hopefully, saving many lives. So with that, I would like to thank Dr. Richard Kuhn for being my PI and for allowing me to do this research in his lab. I'd like to thank some of my Kuhn lab members specifically, primarily Annika Robinson-Hudspeth, Pratik Kumar, and then as well as Andy Miller for their wonderful help in all the things that I've been able to do here. Also, this research was funded through an NRSA F31 predoctoral fellowship, and then was previously funded through T32 training fellowship. But most of all, I want to thank my wonderful wife, Ali Nichols, for her continued support through all this. She's been my sugar mama through grad school, so she makes the money, keeps the lights on. I just do the research, and it's been a fun ride ever since. And so I'm hoping to finish up soon, but I thank you again, Tommy, for allowing me to share some of my work.
Yeah, and thank you, Conrrad. This is such exciting work that you're doing, and it's so. I know you just walked right through some of that as if it were, walk through the park, and I know how much work it really is. So I really appreciate you trying to also bring it down to a level where we can understand and make it attractive for us. Tell me something though, what a journey into research, and what a journey even into science. Do you recall, was it something about science that got you interested at the beginning? Was it because you were in sports? Was it about health? What really was it about science that got you going at the beginning?
Great question. So I think the first time I really got very, very interested in science was I was about 10 years old. And unfortunately, my grandfather did have a brain tumor, and I was really unsure of what was going on, right? Like how can he be so sick? Nothing's wrong with him. He doesn't have a broken bone. I couldn't see anything, right? And so the way my mother explained it to me was, there's some things that we just can't see that can make people really sick, and it takes really smart people to figure it out. And I was like, well, I want to help Grandpa, right? I want to be that guy. And so that was kind of my first interest. But then as I slowly learned more about it, just learned how difficult some problems can be to solve, and that it requires a lot of tenacity, it requires a lot of dedication, it requires a lot of hard work to get it done. I thought that it was a direct parallel. It was a mental parallel to the physical world of playing football, right? And so football's hard. A lot of people get hurt. I got hurt, unfortunately. And so you have to kind of overcome those challenges and hardships in a physical sense. But that's the same idea applied to science for me. So I thought they went hand in hand, right? Everyone's like, oh, you were a scientist football player. That doesn't make sense. And I'm like, to me, it does. To me, it makes perfect sense. And so I think it's a little bit of both, but hopefully that answers your question a bit.
It does. And I think, it's important to acknowledge sometimes when we look back and we have those pivotal moments that change us in one way or in another, and sometimes they are for good, sometimes they're for diseases and things that is beyond our control. Now this environment that you are in at Purdue, this microscope and cryo-EM and the structures of these viruses. Tell us a little bit about that environment, and what is it like to do research with such dangerous viruses? Like, explain to people, are you wearing suits to do this work, or are some of these viruses, can you handle them in a regular lab? I'm sure people are interested to know some of that.
Yeah, absolutely. So it's quite fascinating to work in an environment like this, just with how many unique tools we have here. I think with the whole cryo-EM facility we have, it's not. I mean, we can take samples directly to the microscope with a few help along the way, of course, right? It's not just walk in with it, but We can take samples that we're curious about directly to a microscope and get near atomic resolution images, same day potentially. And so that is very unique, something that I'm very thankful to have because of it's not widespread. A lot of places don't have these resources. And the work that we're able to get done and the turnover that we're able to have in identifying some new mechanisms or new proteins or getting a new structure is quite streamlined. It's obviously you're kind of working on an island in some aspects because it's just you and Richard going on the paper, right? But the skills that you learn to get you to that endpoint are invaluable, in my opinion. Now, on the other side of your question with like working with these pathogens, the nice thing about Hockmeyer Hall and the nice thing about Purdue is that we have those facilities uniquely set up for this specific work. So we have a BSL-2 facility here, meaning that it's a biosafety level 2. We're working in biological safety cabinets, meaning that there's a glass sash in front of us. We have to wear gloves. You're in a lab coat, wearing goggles. We're not in a hazmat suit. Like something like that would be like biosafety level 3. There is a biosafety level 3 facility here, but only a couple people are trained in that. We only work with a few pathogens there. We actually work with SARS-CoV-2 in that facility. But the nice thing about flavies, and I don't want to say nice thing because it's still deadly, right? But they are bloodborne pathogens. And so you can handle them relatively safely, so long as you're not puncturing your skin or ingesting the virus or causing some, like sticking yourself with a needle. Because we do use those equipments and it is quite dangerous. But the training that you get is top notch. The people that you work around are smart. And so it's a good, healthy environment to do some of this work and do it in a safe way. And so unfortunately, it's not something that just anyone can work on, and it does have to require the right facilities. But if those facilities are in place, the research being done is quite streamlined, which is nice.
That's great to hear. And I'm sure that it took a lot of training for you to be able to get to work with the actual stuff, right? Yeah, and I've heard of the extensive training also from the prof sides of things. So it's really interesting. Conrrad, any last words of wisdom that you want to give to people who are thinking about science, who are thinking about scientific research? I really love the way that you put it, that you just sought out research in your undergrad and you tried it out and you liked it and you kept going. And I just wish everybody had that courage and interest and curiosity, natural curiosity that you have, and the qualities of courage to say, yeah, I'll try this out and I'll give it a go. So are there any lasting words of inspiration that you can give to people who are thinking about this?
Absolutely. I will be completely honest and say that research is not for everyone, but it is something that people should try to make sure it's not for them, right? Because it is worthwhile. Because if you do, I know many colleagues of mine who didn't think they would enjoy it as much as they do, and others who thought they would enjoy it more than they do. And the only way that you can figure out where you lie on that spectrum is if you try it. And so if there's any inclination in science or scientific discovery, or if you're interested in how pathogens work, or even just in any field, it doesn't have to be biology, science in general. If you're interested in a particular system and you think you can contribute something, it's worthwhile to try, because you will never know it unless you try. And that's kind of like the whole motto for me, is just, just give it a shot. And if you give it a shot, then you'll find out one way or another. But you'll never know unless you try. So that's all I got. I'm sorry it's not more, but
no, that's fantastic. Thank you for those words of wisdom. Conrrad Nicholls from the Kuhn Lab in Biological Sciences, our Purdue Lecture Hall Series lecturer for today. Thank you again for doing this and teaching us about your world and structural virology and flaviviruses. Thank you, Conrrad.
Thank you. Bye-bye.
Yep. Thank you guys so much. Bye-bye.
Have a great afternoon.
You too. Bye.
Thanks. Bye-bye.